Hi, my name is Ed Dever, and I'm going to be presenting an introduction to dissolved oxygen supporting property measurements from the OI endurance array. I'm at Oregon State University, and I'm a principal investigator and project scientist for the endurance array component of OI. This is an NSF project that's led by Woods Hole Oceanographic Institution with University of Washington and Oregon State University as partner institutions. Dissolved oxygen measurements are found across the Ocean Observatories Initiative. That includes the global arrays, the regional cabled array, the coastal pioneer array, and of course the coastal endurance array. Across all of OI, dissolved oxygen measurements contribute to a number of the science questions that we want to shed some light on using OI, including, what are the role of oceans in the roles of oceans in the carbon cycle, the global carbon cycle? How does severe storm mixing impact physics, chemistry, and biology of oceans? How do climatic signals such as the ENSO, Pacific Decadal Oscillation, and Atlantic Oscillation impact chemistry and biology? How do shelf slope processes structure the physics, chemistry, and biology over continental shelves? And then, the question we're looking at a lot here on the endurance array is, what are the dynamics of hypoxia on continental shelves? And in the left of the slide there, I just represent a couple of the early results from OI. In 2017, Jack Barth et al., or 2018, Jack Barth et al. Wrote a paper on oceanography describing 2017 episodic hypoxic events. Saj Lichtenwaldner at Rutgers University has led a number of these OI data explorations. And then Janice McDonald led the summer 2020 virtual REU project, where several students looked at OI data, including one student who looked at oxygen data off of Washington and compared it to data off of Oregon in the summer of 2017. We also respond to a lot of requests for information from mission-oriented agencies. And that's because hypoxic events over Oregon and Washington can lead to die-offs of organisms, including economically important organisms, such as Dungeness crab right here. So one of the nicest ways to really visualize and understand the dynamics of hypoxia over continental shelves is presented by this J-scope model. And this is a screenshot from a J-scope model taken on the NANUS website, the nanus.org website. And it shows the development of hypoxia. This is a prediction for the summer of 2020 over the Oregon and Washington shelves. And what you can see, this is predicted to be a worse year than average. And you can also see in many cases here, the kind of spatial distribution of hypoxia over the Washington and Oregon shelves. Basically every place inshore of the shelf slope break shown there in the brown line. So the J-scope forecast, it gives data every place. When we're measuring things, of course, we're not able to measure every place, but we are trying to use different types of platforms to complement each other to provide good spatial and temporal coverage. And the three platforms that are part of this webinar series are the coastal surface mooring, glider, and coastal profiler mooring, which has a McLean moored profiler on it. And the moorings, provide really high temporal resolution at fixed sites. They also provide supporting measurements for ecosystem dynamics. The gliders provide spatial resolution between the moorings, and then profilers provide really good vertical resolution at fixed sites. We try to also make sure that these different platforms complement each other by co-locating them. So the profiler mooring is located near a surface mooring. The gliders transit not just between the moorings, but also past the moorings and allow comparisons to be made between those measurements. We have two basic measurement technologies that we're using on OI, coastal surface moorings, profilers, and gliders. The Andorra oxygen optode is used on coastal surface moorings and gliders. It's relatively small, kind of about the size of a pill bottle, so that's one of the reasons it's used on gliders. The Seabird, shown on the bottom there, is the Seabird moored profiler CTD, and the Seabird 43F, which is the oxygen sensor we use on profilers. The reason we use the Seabird 43F on profilers is because it has a faster response than the anaerobic oxygen optode. So as the profiler is traveling up and down, it can respond quickly enough to that profiler movement. As part of OI, we try to do common procedures for data quality assurance and control. That's, every cruise should have the same quality assurance and control procedures, and every array, different organizations, regardless of the organization, we try to use the same procedures. So for example, we all send sensors back to their manufacturers for calibration after deployment. Where it's possible, we do overlapping deployments, in particular of surface moorings. We do shipboard CTDs and physical sampling using common protocols during maintenance cruises. And we employ, community-wide kind of common biofouling mitigation strategies, such as copper tape and anti-fouling paint shown here on a near surface frame. We also try to apply common quality control procedures. So we're doing human in the loop quality control right now with annotation of process data. Some of that data quality control is done by the operators, either it's just kind of routinely or prompted by something an operator might see in real-time data coming back. We also have reviews of data that are prompted by users. And so that's really an important part of OI is getting some community feedback. And if there's a problem with the data set, we try to correct it. And even if we can't correct it, we at least try to make sure that people know about that problem. And we're now working on real-time automated quality control. And I'm going to show a little bit of that quality assurance and control over the next couple of slides right here. So one of the really important things that we've started to do is adopt ultraviolet biofouling mitigation. And this is on surface moorings that are found with sensors in photic zones. And what's shown here in the photo at the left is 2 oxygen optodes. The one that's kind of outlined in blue there is not protected with UV light. The one that's outlined in red has been protected. It's a little tough to show in this, or to see in this picture, but the one that's been protected by the UV light really has shown a significant decrease in the amount of biofouling. This is after a 6-month deployment. And when you look at the time series right here, you can see initially the red and blue time series overlap one another pretty nicely. After about 4 weeks to 6 weeks, you can see here the unprotected one starts to show this enormous increase in oxygen during the day, and that's due to photosynthesis occurring on the biofilm that's now grown on the unprotected optode. And that is outlined here in this box right here, blown up over a shorter period of time. The magenta shows the shortwave radiation, and you can see underlying it is the growth during the day on that unprotected sensor. So this has been a really effective way of mitigating biofouling, and even for deployments of 6 months or longer, as long as this ultraviolet light is working, it can really discourage biofouling and lead to good time series that match, especially on those deployment and recoveries. We can look at the overlap and see that they match. This is work that Craig Rizian's doing, and it's where we're going in terms of automated quality control. So what's shown here are annual and semi-annual harmonics based on data from 2014 to 2019. This actually shows 2 annual cycles, right? So it goes January to January and then another year. And what you can see in the dissolved oxygen plot on the bottom there is during the summer months, that lower oxygen level increases towards the surface. And this is water that is then upwelled near the coast and contributes to the development of hypoxia, especially after organisms grow in this nutrient-rich water and then fall out of the water column and decay and drive the oxygen levels down even more. So in the long run, what we're going to be doing is we're going to be using these seasonal cycles to then start to look at anomalies from those seasonal cycles to alert people when there are interesting events occurring, or possibly if there's a problem with the data. In the short term, some of the things we're doing is we're doing additional kind of human in loop quality control. And we've got a poster that's submitted together with our colleagues at the Regional Cabled Array for the fall 2020 AGU. And some of the things that we're doing is we're going to be doing more comparisons of shipboard CTD dissolved oxygen data to mooring time series. That what's shown here is this is just all the bottle samples and all the shipboard CTD samples from coastal endurance cruises on a variety of Udall ships. They generally line up pretty well, and that's going to allow us to use the shipboard CTD measurements to really match up in temperature salinity space the oxygen measurements that we're taking. So we'll get better better comparisons to dissolved oxygen with good kind of validated shipboard CTD dissolved oxygen measurements. We're also going to be comparing mooring time series together with profiler and glider measurements, as I alluded to earlier. And we're going to be adding some additional platforms to the platforms that we're doing quality control on. So right now we're looking at moorings, gliders, and profilers. We're going to add regional cabled array oxygen measurements, as well as the uncabled coastal surface piercing profiler to our quality control presentation in fall 2020. And then in a little bit longer term, we'll be implementing those automated quality control checks from using the Quartile methodology. And then finally, there's a new data portal in development by Axiom Data Science, which is going to roll out in next month, October 2020. So part 2 of this series of talks, Chris Wenger is going to talk about mooring time series. In part 3, Stuart Pierce is going to talk about glider time series, and Craig Rizian will talk about the Washington Offshore Profiler mooring time series in part 4. As part of each one of these talks, they're going to review data from the platforms listed above, as well as kind of focus on summer 2020 measurements, that is recent measurements that people may still be quite interested in understanding. And we're going to focus on data rather than data access methods here. So there will be some description of data access methods, but please, if you're having trouble accessing data, if you'd like a tutorial on accessing the data that's important to you, let us know and we can schedule events either tomorrow, September 10th, or later on. And then we're going to be presenting these dissolved oxygen data with some context from related property measurements such as wind and water temperature. Having said that, there's a lot of other physical, chemical, and biological time series that we don't have the time to present. For each platform, the measurements, the other types of measurements will be described briefly. And for what it's worth, we have several poster presentations that describe some of these measurements in more detail, including meteorology, chlorophyll fluorescence, and air CpCO2 measurements. And if you're interested in looking at some of those posters, let us know and we'll get them in your hands. With that, I'll end and thank you very much.